Hello and welcome to all my dear student. Very good morning to all of you. Again, I would like to welcome all of you in this science lecture. So today we are going to discuss the in-text questions as well as the exercise questions. Right? So let us start today's session. In our previous lecture we have ended the unit 5, the fundamental unit of life, cell. Okay, so first intake question, page number 59. This is about the discovery of cells. Who discovered cells and how? Cells were discovered in 1665 by English botanist Robert Hooke. The botany scientist Robert Hooke discovered the cell. He used the primitive microscope to observe cells in cork slice. Cork यानी कि जो वुड का एक भाग होता है उसके अंदर उसने सेल्स को ऑब्जर्व किया था. ओके, चलिए. वेरी सिंपल क्वेश्चन इज दैट. सेकंड, व्हाई इज द सेल कॉल्ड द स्ट्रक्चरल एंड फंक्शनल यूनिट ऑफ लाइफ? बिकॉज द सेल्स आर अ बिल्डिंग ब्लॉक ऑफ द लाइफ. सेल made the tissue and all the body organs are made up by the cell. So the basic constitute of any our body organ is cell. So here it is mentioned in another different kind of language, but the answer is same. See, cell constitute various components of plants and animals. A cell is the smallest unit of life and is capable of all living functions. It is a building blocks of life. This is the reason while cells are referred to as the basic structural and functional units of life. Okay? All cells vary in their shape, size and activity they perform. So these are some of the important characteristics of the cell, and because of these characteristics the cell is called the structural and functional unit of life. Moving towards the another question. Question number 1. How do substances like CO2 and water move in and out of the cell? Discuss. So first of all, I already told you in our previous lectures that the exchange of gaseous substance and the liquid substance is through the two processes. Which are they? The first, in which the gaseous exchange takes place, the process is known as diffusion. The second, in which the liquid matter is exchanged, and the process is known as osmosis process. And this all entry and exit of the external matter is made with the help of selectively permeable membrane. Okay, so in this way the CO2 and the water move in and out of the cell. This is the all theory that is written here. You should also write that theory. Okay, higher concentration of CO2 inside the cell, lower concentration of CO2 outside the cell. So in this way the CO2 move from inside the cell to outside the cell. So this is just a simple example that you should write in your own language. It is not necessary to write in this language, but you can write the answer of the question in your own language. Question number 2. Why is the plasma membrane called a selectively permeable membrane? The reason that why plasma membrane is called selectively permeable membrane, because it regulates the movement of the substances in and out of the cell. Means certain kind of substances or objects only allowed to enter inside the cell. Each and every substance are not allowed to enter inside the cell, and this means the plasma membrane allows the entry of only some substances and prevents the movement of some other materials. That is why it is called selectively permeable membrane. I hope that you understand it very well. So ये question number 1 इन टेक्स्ट का पेज नंबर 63 के ऊपर का कि जो आपको पूछा गया था पीरियोडिक एग्जामिनेशन के अंदर, प्रोकैरियोटिक सेल एंड यूकैरियोटिक सेल. तो प्रोकैरियोटिक सेल के अंदर न्यूक्लियर रीजन जो होता है इट इज नॉट वेल डिफाइंड. ओके, एंड नन एज nucleoid, poorly defined because of absence of nuclear membrane, and it is known as nucleoid, right? देन आफ्टर फोर्थ नंबर के अंदर यहाँ पे in the prokaryotic cell, membrane-bound cell organelles absent, and here it is present, like mitochondria, Golgi apparatus, plastids, vacuoles, etc. Okay? चलिए आगे बढ़ते हैं. Page number 65 के ऊपर question number 1. Can you name the two organelles we have studied that contain their own genetic material? क्या आप ऐसे दो सेल ऑर्गेनल के नाम दे सकते हो कि जिसके अपने जेनेटिक मटेरियल होते हैं? ठीक है? So yes, we can give. First is mitochondria and plastids are the two organelles that contain their own genetic material. जब हम पढ़ रहे थे, आप प्रीवियस लेक्चर को भी रिवाइंड करके देख सकते हो कि मैंने उसमें बताया हुआ था कि प्लास्टिड जो है तो वो सेम माइटोकॉन्ड्रिया के जैसे होते हैं कि जिसके पास अपने जेनेटिक मटेरियल्स होते हैं. ठीक है, डीएनए और रिबोसोम्स अपने अपने होते हैं. इट मीन्स द जेनेटिक मटेरियल. क्वेश्चन नंबर 2. If the organization of a cell is destroyed due to some physical or chemical influence, what will happen? अगर सेल के ऑर्गेनाइजेशन के अंदर कोई फिजिकल या तो केमिकल इन्फ्लुएंस से डिस्ट्रॉय हो जाए सेल, तो क्या होगा? लेट अस डिस्कस. सेल इज द स्मॉलेस्ट यूनिट ऑफ लाइफ व्हिच इज कैपेबल ऑफ ऑल लिविंग फंक्शंस. If the organization of a cell is destroyed due to some physical or chemical influence, then the ability of the cell to perform all living functions such as respiration, nutrition, excretion, etc. would be affected. So as we know that the cell is a basic fundamental unit of life. It is a basic structural member of any body organ. So if the cell is destroyed by physical or chemical reason, their function is directly affected. And if the function of the cell is affected, then it will affect the whole body organ like heart, leg, kidney, stomach. So these are the some body organs in which the prime constitute or the basic constitute is cell. So it can also affect the whole body organ. Third question, why are lysosomes known as suicide bags? So when we are discussing the lysosomes, I already told you in a detail that the why lysosome is called as suicide bags, because when the cell is worn out or it is dead, then at that time lysosome secretes some kind of chemical and the lysosome solely can absorb the cell. Self-destruction of a cell by releasing the digestive enzymes within the cells. Okay, and that is why it is called as suicide bags. When are proteins synthesized inside the cells? Ribosomes are the site of protein synthesis. यानी कि प्रोटीन कहाँ पे बनता है? सिंथेसिस का मतलब होता है बनना. रिबोसोम्स की जो किसके साथ अटैच्ड होते हैं? एंडोप्लाज्मिक रेटिकुलम. मैंने दिखाया था ना, वहाँ पे ऐसे एक पार्ट था और उसके इर्द गिर्द ऐसे छोटे छोटे पार्टिकल्स होते थे. समझ में आया? Ribosomes are very small structures found either in a free state, suspended in the cytoplasm, or attached to the surface of endoplasmic reticulum, जैसे मैंने आपको बताया. So ribosomes के पास क्या क्या होता है? Ribonucleic acid and proteins. So at that point, at that site, the protein is synthesized. Okay, so these are the in-text question that you should write in your fair book. After this, exercises. Exercise के अंदर question number 1. Make a comparison to write down ways in which plant cells are different from the animal cell. So here they are saying that to differentiate between plant cell and animal cells. So let us differentiate. First, the size is small, animal cell, and the larger in the size than the animal cell. Cell wall is absent inside the animal cell because it has a plasma membrane. In a plant cell, plasma membrane of plant cells is surrounded by rigid cell wall of cellulose. Okay, so plant cell is also have cell wall with the plasma membrane. Animal cell has no plastids while the plant cells are having plastids. Vacuoles in animal cell are small, but in the plant cells it is permanent and large size vacuoles are there. Animal cells have single highly complex and prominent Golgi apparatus, while plant cells have many simpler units of Golgi apparatus. Means here a single Golgi apparatus is present, while inside the plant cell many small Golgi apparatus are present. Animal cells have centrosome and centrioles, and plant cells लेक ये हमारे पढ़ने में सिलेबस में नहीं आया, इसलिए इस टॉपिक को आप निकाल सकते हो. 5 पॉइंट काफ़ी है. Question number 2. How is prokaryotic cell different from the eukaryotic cell? Let me tell you. So यहाँ पे जो आपका जो वो टेबल था उस टेबल को ही आप कंसिडरेशन में ले सकते हो. ठीक है, एग्जामिनेशन में दिया गया था. सेल साइज इज जनरली स्मॉल, सेल साइज इज जनरली लार्ज, nuclear region is called nucleoid and it is not surrounded by nuclear membrane and it is surrounded by the nuclear membrane. Single chromosome is present in the prokaryotic cell, more than one chromosome is present inside the eukaryotic cell. Nucleoles is absent while it is present. Membrane bound cell organelles are absent while the membranes bound cell organelles present. Okay, chaliye. So these are the differences between the prokaryotic and the eukaryotic cell. Question number 3. What would happen if the plasma membrane ruptures or breaks down? As we know that the plasma membrane is a selectively permeable membrane, and selectively permeable membrane allows certain objects entry and exit. So if it ruptures or breaks down, constant internal chemical composition of the cell will be lost and it will not be able to perform its basic functions. So such a cell with ruptured plasma membrane is killed. Means the plasma membrane is not present, that at that time the cell will itself killed. So the simple understanding is that whenever any of this cell organelle, if it is whether it is a plasma membrane or it is a cell wall, if it is rupture or breakdowns, see one thing, they are not able to perform their specific function. So cell is also not performing that function. So cell itself killed. Question number 4. What would happen to the life of cell if there is no Golgi apparatus? So if the no Golgi apparatus is there, then the material synthesized in the ER are stored, sorted and modified, packaged and dispatched to various targets inside and outside the cell through the Golgi apparatus specs. So the Golgi apparatus is a transportation device of the cell, which is transport, modify, pack, store and sort the various materials. Okay, so that transportation is not takes place if the Golgi apparatus is not there. Okay, so Golgi apparatus, if it is not involved in the formation of cell, then the cell is not no more present. Sometimes the Golgi apparatus is also included in the formation of cell. So if it is not present, then the cell will not form. Okay. Question number 5. Which organelle is known as the powerhouse of cell? Why? As we know that the mitochondria is called as the powerhouse of the cell because they contain enzymes that are needed for stepwise oxidation of foodstuffs, like to digest or to convert the complex substances into the simpler one and produce the energy. That organelle is known as mitochondria. Okay, so that is why it is called as the powerhouse of the cell. I already discussed in the detail when we are in the lecture section. Question number 6. Where do the lipids and protein constituting the cell membrane get synthesized? So there are two sites, RER and SER, rough endoplasmic reticulum and smooth endoplasmic reticulum. So rough reticulum synthesize protein while the smooth reticulum synthesize the lipid. Okay, you should remember it. How does amoeba obtain its food? Amoeba is a flexible cell membrane. It enables amoeba to engulf in food by the process called endocytosis. So by the endocytosis process, amoeba obtain its food. Okay, चलिए. What is osmosis? Osmosis process क्या है? So osmosis is a diffusion of water or solvent through a semi-permeable membrane from a solution of lower concentration of solute to a solution of a higher concentration of solute. Okay, So it is nothing but the movement of water from higher concentrated region to the lower concentrated region, in which the water is transported from or to the cell. Understood or not? इस प्रोसेस के बारे में मैंने आपको डिटेल में बता दिया है. तो ये है आपके एक्सरसाइज के क्वेश्चन. यहां तक रखते हैं आज का लेक्चर. नेक्स्ट लेक्चर में नया यूनिट स्टार्ट करेंगे, यूनिट नंबर 8 मोशन. थैंक यू.